We're going to go ahead and get started with today's lab, the first of our series called Lab Before Time, where we are kind of looking at prehistory, at time before time. So also known as deep time, I think Max referred to it as. So today we're going to be talking about primordial soup, is what it's often called. Now, primordial soup does not come in a can. It's just sort of an idea about how life first started on Earth. So if we go back more than thousands, more than hundreds of thousands, more than millions, more than billions of years to around maybe 3.5 billion years ago, that's when scientists, that's how far scientists have been able to date kind of the earliest fossils of life. And it's a certain kind of life. It's a type of life that's very, very small, known as bacteria. What do you guys know about bacteria? Or maybe what does it make you think of? What do you know about bacteria? Because it's something, bacteria is something that's not just ancient, it's also still all around us today. What do you think?
Good point.
Go
Somebody
ahead.
said it can make you sick. That's pretty good. Is bacteria big or small? Big or small?
What do you guys think? Oh yeah, you got it. It's tiny. Yeah, it's super small, right? So bacteria is something really small. It's still, we can still find kind of fossil traces of it. And kind of the earliest we're thinking is about maybe 3.5, maybe 3.7 billion years ago. And we call this idea of kind of life emerging from this concoction of stuff, probably in a body of water. We call that primordial soup, is that idea. So today we are going to be making a sort of soup, although it's not going to be very primordial. We're not going to be making something from nothing. We're not going to be creating life here in this lab, although it has been done on some scale before. There's a famous experiment where we took a whole bunch of different inorganic, non-living chemicals and put them together, had an energy source, a heat source, and in the process of a couple of days, there were organic molecules that came out of it. These random non-living chemicals did tend to form together into some of the building blocks of cells. That's still a lot between, right, like some building blocks of cells and whole living organisms, but it's still a start. Anyway, we're not going to be mixing up, we're not going to be creating living molecules today so much as we are going to be creating our own little atmosphere for life. We're in some ways making an early Earth using some materials that I think you can find pretty easily. I'll go ahead and show you what I've got. Are any of you doing this experiment with us? What do you think? While you let me know, I'm going to show you what I have because I have a jar. I've got my jar here. You can use anything for your kind of, for your model of early Earth. You can use anything that's kind of like a cylinder like this, kind of tall, and anything that's clear so that it lets light in. So you could use an empty water bottle, you could use a soda bottle, anything like that. I have a jar. Oh, cool. It sounds like we have some people doing it with us, and then some may do it later. That's awesome. So we have this jar. It's going to be our early Earth model, sort of. And it's also got its own name, right? It's called a Vinogradsky column. Try saying that 5 times fast. So our Vinogradsky column is just a collection of different bacteria, different microbes, things that are teeny, teeny, tiny, and they look really interesting once we give them a little bit of time. So it all starts with this jar. The other thing that you're going to need is dirt. Now I've got plenty of dirt in here. This is some dirt that I got from kind of the banks of Ellerby Creek. And I've added some water so it's kind of muddy right now. There's a lot of other stuff in here like pine needles and roots and kind of some different stuff. So it's all some good dirt, good soil from outside. I've also got some water. And this water is also from Ellerby Creek. Any water from outside will be perfect to use for this experiment. You can also use water from the tap, but it has to be dechlorinated. That just means when you put it from the tap into your container, you have to let it of, you have the right water to start your experiment. So we're saying we have dirt, we're saying we have water, we've got some sort of container, and we've got some other pieces. I've got a piece of paper towel. I only have like a half here. And this paper towel is actually going to serve as a kind of fuel for what we're going to put in this jar. We'll talk about that in just a minute. Well, I've got a little bit of paper towel. You could also use any kind of paper, really. Newspaper is another good one. I've also got a source of sulfur. So I chose to use some Epsom salts. And you can see in Epsom salt, there is magnesium sulfate. So that sulfur, that chemical, is going to really help us out. It's going to be my sulfur source. And then I want to have a source of carbon. There's a lot of different things I could use for that. I could use a bunch of pine needles or dead leaves. I could use even more paper. I have a little bit of cornstarch just because I thought that was an interesting idea. And I think we're gonna have that be our carbon source for this time. Last but not least, I do have just a few handy tools with me, like a spoon for scooping. I have scissors just in case you have paper that you might need to cut into strips. I think I'm going to be okay ripping mine. And of course, I've got some plastic wrap for later. And I've got a towel for when I inevitably spill something. So we're going to be making some sort of primordial-ish soup inside of our jar or inside of our container. And we're calling that a Vinogradsky column. That is a miniature little environment in here that has many different kinds of bacteria. Some of the bacteria that can be found in this column that we're going to make is the same bacteria that can be found long ago, like we were talking about in deep time, some of the very first bacteria to live on the planet Earth. So we're going to go ahead and start making here. I'm going to switch to Experiment Cam.
I love Experiment Cam.
I'm so glad you love Experiment Cam, and thank you for being here, Max.
I'm so happy to be here. So I have a couple of questions, maybe for you, Peregrine, but also while you're getting set up, maybe also for everybody else who's joining us. First of all, why do we call it a Vinogradsky column?
Oh.
Anybody can take a guess at that. Why Vinogradsky column? And there are two words there. You can take a crack at either one of them. What do you think?
While we're thinking about that, what I'm going to do is going to be the first step of our experiment. We're going to just start ripping up some small pieces of paper towel while we're thinking.
Love that.
Oh,
somebody said maybe because it's a column. So what is a column? What does that word column mean? Or perhaps what direction do we think about columns in?
I think about a column going up and down.
I also think about a column going up and down. So I think about the architecture, that piece that holds up a building because it's very tall. So we are going to be making a column. We're making something like the architecture where it's going to go from the bottom and vertically, using somebody else's really excellent word here, up to the top. So what we're going to build is something that's going to look different down here at the bottom than it does up here at the top. And of course, that Vinogradsky piece is because it was first sort of made or first sort of coined by somebody named Vinogradsky.
Our guy, Sergei Vinogradsky, this is a very cool idea, right, about observing microbes, observing bacteria in an environment where they all live together. Instead of it just being kind of one of them in a petri dish and observing that under a microscope, the idea here is to see an entire ecosystem, an entire soup can, right? So I've got my column here. I've got some ripped up paper towel or any ripped up like paper that you have. So I'm going to go ahead and put it in here. And what this does is this is going to be some cellulose, right? We know that paper is made from plant fibers, often made right from trees. So this is going to be a good source of kind of like a fuel for all of the things that are going to be living inside of our column, something for them to eat. So we have a little bit of cellulose and a little bit of creek water in here. And now I'm going to add a little bit of my dirt. So I'm going to go ahead. My dirt is already kind of in mud form, so I'm just going to scoop it using my spoon It was a lot of fun earlier in Little Lab when we did this experiment. We mushed up a lot of our mud with our hands, which was super fun. And if that's something that you all are interested in doing, I would love for you to get your hands dirty. Because a lot of science isn't done inside of a sterile lab. A lot of it's done out by the sides of creeks or with lots of, like with using mud and not necessarily always having, having gloves on. I know I don't have any gloves on today. So I've got a bunch of scoops of mud here. I'm gonna do maybe one more big scoop. There we go, that looks pretty good. Now it looks like I've got a bunch of muddy water, which I do, and I'm going to add those sources of sulfur and my source of carbon. So I've got my Epsom salt here, and I'm just gonna pour mine in because it comes with this handy spout, but really if we do about 4 spoonfuls, that should be about enough, but you could always do a little bit more. That'll affect the kind of bacteria that are able to grow. A lot of the bacteria that we're gonna be growing in here are bacteria that like to eat sulfur. So this is another one of those fuel. All right, so I'm pouring that in here, about 4 spoonfuls, give or take. It's going to get in here. Another source of sulfur that you could find around your house would be like the yolk of an egg, although I've heard that those Vinogradsky columns tend to get really smelly because of the anaerobic bacteria that like to break it down. So we've got a little bit of our sulfur source in there, and now I'm going to add my carbon source, and I'm going to use a little bit of cornstarch here. I've also heard you can use oatmeal, right? As well as other kind of more natural sources, like we could use leaves, we could use pine needles, stuff like that. So I'm gonna stir this up some more. And I don't know about you, Max, but I don't see any big reaction happening. What about you?
You know, I don't either, Peregrine.
So this isn't something that we're going to be able to see immediately. It's not even something that we're going to be able to see in a couple of hours. The kind of growth that's going to happen in here is going to take a while. We might not even be seeing the first signs of life, at least to our eyes, right? For maybe a week or so, maybe a couple of days if we have a really good source of nutrients in our column. But for mine, I bet I won't see any colors or anything interesting looking for a little while, probably for a few weeks. So I've got all of my stuff in here. I've got my soup, and I'm going to add a little bit more water here at the top. There we go. Just adding a little bit of water in the top. So this makes for a pretty good column. You can already see something starting to happen, right? This is just the water settling a little bit. So we kind of have a layer between our dirt, our soil, and the top of it, the top of our column there. And what we're creating is an environment where down here, there is not a lot of oxygen. Right, so oxygen is all around us. It's what we need, what we breathe in from the air all around us, what our bodies need. But some types of microorganisms, some sorts of bacteria can grow without a lot of oxygen. So we're going to see more of those toward the bottom. We would call those our anaerobic bacteria. Oh, sounds like Max is having a little bit of technical trouble, but he'll be right back with us. So at the bottom of our column is where we're going to have those bacteria that don't like to breathe or don't like to use oxygen, right? Whereas at the top, you'll have the kind of bacteria that like light a little bit better and that also can use a little bit more oxygen. So that's going to change the way that this whole thing looks. And I wanted to show you all some pictures of how these things look a couple weeks on, because it looks a lot different than just a column of dirt. So I'm going to go ahead and share my screen. Not this one. I'm going to use this one. Perfect. So these are some pictures of different Vinogradsky columns kind of before they've had a lot of time to develop, but there's something that over time kind of becomes something really beautiful and gorgeous, because a lot of these different bacterial groups, bacterial colonies, get to be a lot of different colors, right? You can see that a lot of times there's some green near the top, there's kind of some reddish, there's some purplish and pinkish. There's all these really beautiful colors. Here's another ones of these here. There's also lots of different textures and almost like designs, some cool little squiggles and stuff. I found this kind of moving picture that shows some of these bacterial colonies developing. This is sped up, I think it said several thousands of times, right? So this isn't something we could see just with our bare eyes, but over time, this is how it develops. Max, we're so glad to have you back.
I'm so glad to be back. Sorry about that. I don't know what happened.
We're glad to have you. We were just taking a look at how our Vinogradsky columns might look in a couple of weeks. But right now, it just kind of looks like this. What we might do just to finish it off is we're going to take our plastic wrap and we're going to put it over the lid of our jar just to kind of create sort of a closed environment. You can just leave it like that, but I might put a rubber band over mine. And now my soup container is complete. I'm going to take it back to my experiment cam. So I've got my column all set up here. It has a source of cellulose. It has a source of carbon. It has a source of sulfur. It's got kind of this water and this mud that already have small amounts of those bacteria already present. So what they're going to do is they're going to eat all of the stuff they like in there, and they're going to create their own environment where they live together. There's a certain kind of bacteria that we'll probably start seeing in the first few weeks. They're probably some of the first bacteria that we'll start being able to see with our eyes. And they're going to be on the top here. And this is the reason why I wanted to ask my friend Max in, because this is his favorite kind of bacteria. So I wanted to pass that over to you, Max. What kind of stuff will we see on the top?
Well, first of all, everybody should come up with their favorite type of bacteria. It's important that everybody have one, right? And this goes to why we would do an experiment like making a Vinogradsky column in a lab called Lab Before Time, because like we started out talking about, we are talking about deep time. We're talking about time that happened so long ago that it's kind of even hard to think about. So not thousands, not even millions, but billions of years ago. And if you think that far back into the past, the world obviously looked like a pretty different place. And I don't even mean that, the people back then didn't have cell phones or whatever. When we think about life that far back, all life was about the size of bacteria. It was all one-celled organisms. And there's another really fascinating thing that was going on then, which has to do with the atmosphere of the Earth, a protective layer of gases around the Earth. Before we go any further, let's all just take a deep breath together. Ready? 3, 2, 1. Okay. What did you just breathe in, by the way? What did you breathe in?
We may have even mentioned it earlier. What do you think, friends?
What did you just breathe in? So our atmosphere now is pretty well set up, like some of y'all are saying, for us to breathe. In fact, most living things on Earth need the basic chemicals that we have in the atmosphere now to survive. And most of what you actually just breathed in is nitrogen. Most of our atmosphere is made up of nitrogen. But really importantly, the air that we breathe has oxygen in it. It's really, really, really important to us. Back 3.5, 3.2 billion years ago, we only had about 1% of the oxygen in the atmosphere that there was right now. Most of the air in the atmosphere was other stuff. It was other chemicals. And all of those one-celled organisms that lived back then absolutely loved that stuff. They needed all of those other things to survive instead. And along came this really, really interesting thing that, again, you'll be able to find in your own little early Earth model in your Vinogradsky column up near the top. And that is something called cyanobacteria. Try saying that 3 times fast. Cyanobacteria, I think, is so cool because cyanobacteria basically made the world that we live in now. As the name sort of suggests, cyanobacteria is a bacteria, but it's one of the very first things that could photosynthesize, turn sunlight into its food, and then photosynthesize in a way where it got rid of oxygen. So like modern day plants, like a lot of what you're seeing around me, it got rid of oxygen as a byproduct. As more and more cyanobacteria started to grow here on Earth, what happened? Remember, our atmosphere back then did not have very much oxygen in it, but with more cyanobacteria came more oxygen, which sounds great to us because, again, we need oxygen to survive. But all of those teeny tiny little one-celled organisms that lived back then, what do you think they thought of all that oxygen?
These would be the same ones that would live near the bottom of our Vinogradsky column that like to eat stuff like sulfur, but aren't fans of oxygen.
Unfortunately for all of them, the ones that you're not going to find in those Vinogradsky column are ones that no longer exist on Earth. A lot of things went extinct. We call this the Great Oxygen Crisis, and it's one of the very first and largest extinction crises here on Earth. Because so much oxygen kind of suddenly or very quickly ended up in the atmosphere, a lot of things that found oxygen totally toxic to them could no longer survive. So lots and lots and lots of them went extinct. But with that extinction came an opportunity. And like I said before, cyanobacteria basically made the world we live in because without that oxygen, there wouldn't be a world that could support multicellular, that's more complex life than teeny tiny little bacteria, to survive. So because of cyanobacteria, you end up with pretty much all of the rest of life on Earth that comes afterwards. I think this is really neat because cyanobacteria is one of the things you're going to find near the top of your Vinogradsky column. And like how it did 3.5 billion years ago, it's going to create oxygen inside of there. But like Peregrine said, the reason that we see this column is because that oxygen is going to be up here near the top. Down there near the bottom where you've created these kind of dense layers that it's hard for oxygen to get through, you've instead made a home for things that don't need or perhaps don't even like oxygen that much. Now, I love this lab, and I think this is so cool because we're not creating a real primordial soup and that we're not creating life out of a bunch of different ingredients. Instead, we're using those ingredients to create kind of a little home or kind of a little apartment complex maybe for a few different species of little things. And if you've joined us for the lab before, this is a lot like a few other labs that we've done where we've made little homes for other things. You may remember this is very similar to what we did with our terrariums. Here's mine again, and you can take another look inside. And if you look in there for long enough, guess what you will find in there?
Is there even some cyanobacteria inside of your terrarium?
There is even cyanobacteria inside of my terrarium. And if you look along the side here, you've actually got a sort of small Vinogradsky column. And if you go even further back in lab history, you might remember we did this also with our sourdough starters. We also made a little home for little things that like to live inside of this mixture of flour and water. This is a really similar process. Now, I encourage you not to try to eat your Vinogradsky column because it's going to probably smell a little bit bad and it wouldn't be good for you and, all of that stuff. But in a really, really similar way to our sourdough starters and our terrariums, what we're doing is we're making a little home for microbes, for life on Earth that survives in these really, really interesting ways that we sometimes don't get to see or think about. I'll end my talk about cyanobacteria, my very favorite bacteria, by saying that this bacteria is not just one species. There's some types that are toxic, that turn the water different colors, that you would not want to get in, and that fish don't want to be in. There's other types that we actually eat. In fact, if you've ever eaten like blue gum or candy, that coloring is made using cyanobacteria. So it's still a very, very, very common bacteria that we find here on Earth outside of our Vinogradsky columns too. I just think this is such a really, really cool way to exercise a little bit of scientific patience and to get our hands dirty and to get to observe things changing after you've added a bunch of initial ingredients, which is such a lovely way for me to think about the way that science processes work.
So what we're going to be doing is in the weeks following, I know that I am going to be watching my Vinogradsky column. I'm going to be putting it kind of near a window. I don't want it to get a lot of direct sunlight, but I want it to have plenty of light because there's definitely things in there like cyanobacteria that like to, that need the sun, that photosynthesize, as well as things that don't like the sun as much. So I wonder if the level of light might affect the different colors and the types of bacteria that I'll see. I think that's an interesting hypothesis to explore. So this is something that I think I'd look to check every morning. I know after a couple of weeks, I'll start to see it look like a little bit more than just kind of like a mud puddle. But I hope that you all will continue to watch your Vinogradsky column as well. And that if you see something really cool, you might share it with us. If you'd like to take a picture and put it on social media and tag it, tag the museum and then tag it Lab at Home, we would love to be able to see the kinds of cool stuff that your Vinogradsky column made, because they're all going to be a little bit different. We're all using slightly different ingredients. Probably we're getting soil from a different place. I'm pretty sure you guys aren't getting soil from the exact same place as me anyway. So I'd be really interested to see. I hope that you start to see some interesting colors and some interesting shapes and textures. I know that I am looking forward to seeing some as well. So that's our kind of going back as far in time as we're able to, right? But next time we are going to have a lab about another prehistoric creature or group of creatures. We're going to be talking about dinosaurs, everyone's favorite. Specifically, we're going to be talking about dinosaurs and the ways they might have sounded. We're going to be experimenting with sounds. This is a great question. Does it need to be in a warm place or does it not really matter? I think if you have this out and the water freezes, probably that's going to inhibit growth a little bit. I don't think it needs to be kept warm, but probably as long as it's not frozen, I would say that's probably pretty good. In general, the slightly warmer it is, I think the more / faster it's going to grow. But I think it would be pretty hard to keep these guys down. I think even frozen, they might be able to grow a little bit, but that is a great question.
And remember, they've lived on Earth for 3.5 billion years. Some of them are the most resilient life forms that we've ever seen.
Life finds a way. So slightly warm, I think, is our uptake, not freezing anyway. I hope you might join us next week to talk about dinosaurs. We're going to have one of our friends who is a paleobiologist who's going to talk to us a little bit about her interests. She has a lot of cool knowledge about dinosaurs. I hope that you guys might join us to share your cool knowledge about dinosaurs, because it's a subject I don't know much about and I'm really excited to learn more about. So friends, I hope we'll see you next time. Thank you so much for joining us. Thank you, Max, for being here to talk about your favorite bacteria. And I hope you all have a great time observing your awesome columns. Thanks so much, friends.
Bye, everybody.
I hope we'll see you next time. See you later, friends.